Good afternoon. It's a great pleasure to welcome Ilya Ruvinsky from the Department of Ecology and Evolution at the University of Chicago. Ilya comes from Novosibirsk, where this is a balmy day today. He then did his PhD with Lee Silver, someone that many of us know, at Princeton, and then went to Gary Ruvkin's laboratory, where he discovered the joy of C. elegans down at Harvard Medical School, actually Wellman Building. And since then, Ilya's been at the University of Chicago, where today he'll tell us just wonderful studies on how C. elegans tells us about genes, gene structure, and gene evolution.
Thank you, Rick. No, I think I have it already. Am I now free to turn this slide off?
I think you're free to turn that slide off.
Very good.
That's the advertisement.
Yes, Coke will be distributed after the talk. All right. All right, Rick, thanks very much for this kind of invitation and the introduction. What I'm going to try to do today is tell you two and a half stories that represent vignettes of how we're thinking about approaching evolution of gene regulation. And one of the questions you might ask yourselves is, why am I talking in the Department of Molecular Biology if I am in the Department of Ecology and Evolution? So I was trained as a molecular biologist, but by intellectual inclination, by the questions that I'm interested in, I want to study evolution by applying the tools of molecular biology and the reasoning of molecular biology to long-standing questions, long-standing evolutionary questions. So what I'm going to tell you today is about how we study evolution of gene regulation. And it's one of those things that could mean three different things to two different people. Specifically, when I say evolution of gene regulation, what I mean is that I'm interested in understanding specific functional consequences of changes in either the cis or the trans components that affect gene expression. And so there are three broad categories of reasons why it might be interesting to study a problem like this. One is that it goes, obviously, gene regulation is important in development. Development leads to phenotypes. Phenotypes is what changes in evolution. Therefore, if you want to understand something about evolution, you want to understand how gene regulation changes. So that's one big motivating factor. Another, of course, is help with comparative genomics. Now, the promise of comparative genomics has been from the outset that we're going to sequence a lot of genomes, we're going to look at them, we're going to compare them in some intelligent ways, and all of a sudden, from these billions of letters of nucleotides will spring out messages, functional sequences that would otherwise be very difficult to infer. And so understanding the dynamic of how genomes change over time, how gene regulation in particular changes over time, is an important component to that endeavor. From a more evolutionary perspective, transcription, because we understand a fair bit about it, provides an interesting model system in which to investigate how complex systems evolve. And so motivated by all three of these principles, we've embarked on a series of studies that I will describe today. It's been roughly 20 years now since the following observations have been made. People started cloning highly conserved genes that determined various elements of development. And what they found, by and large, was that even in very distantly related organisms, same genes were expressed in patterns that could be interpreted as being conserved. And here's one of the more notorious examples of PAX6 and PAX6 related genes being expressed in photoreceptor neurons in a whole variety of multicellular animals. And so the question arose early on is, what is it that makes these genes be expressed in fairly conserved patterns for very long periods of evolutionary time? Well, so you can come up with one set of ideas, and sort of a really straightforward hypothesis would be that fundamentally cis-regulatory sequences and gene regulatory apparatus is highly conserved. Sequences, of course, change, but perhaps not in ways that are meaningful to their function. And therefore, let's say if you consider two different worms that descended from a common ancestor, even though some changes have occurred, they were inconsequential functionally. And therefore, patterns of gene expression have been retained by these two descendants. An alternative hypothesis is that there has indeed been a fair bit of functional change. That is, this red bit of DNA and the blue bit of DNA are not functionally equivalent. And yet somehow systems have changed, evolved concomitantly to these changes to accommodate for the differences and to somehow buffer the effects of mutations that were being introduced. And so it's with this in mind that we're going to talk about the first couple of sets of experiments for the majority of the talk today. The model system that we're using is nematodes, and it will become clear in a minute why nematodes. We know a lot about their development, but we can also have very precise readouts of gene expression patterns. And I'm going to talk about C. elegans and a fairly closely related species, C. briggsy, as well as some others. And these are, although morphologically very similar, separated by a fair bit of evolutionary distance in terms of mammalian, to compare to mammalian divergences. This is something like orders of mammals. And for those of you in the audience who are fly aficionados, it is a little more distant, this pair of species, a little more distant than Drosophila melanogaster and Drosophila pseudoobscura. And this is what's illustrated in the slide. You can normalize the amount of divergence between the species that we're considering over here, and you can see that it's sort of divergence between distantly related flies or divergence between, say, mammals and, or placental mammals and marsupials. The technique that we're going to talk about for the majority of the talk is very straightforward. A piece of DNA that we're interrogating for gene regulatory function is fused to GFP. We make transgenic worms, and then we'll look at patterns of gene expression. All right, so let's dive in the meat of the story. For somewhat historical reasons, but also because of the beautiful expression pattern, a gene called UNC47 was selected for study. This gene, UNC47, is expressed in a pattern where 6 cells express in the head, 19 cells in the ventral cord, they're called D-type neurons, and 1 post-anal cell, DVB. These are 26 GABAergic neurons in C. elegans. These are all GABAergic neurons, and these are all GABAergic neurons that this worm has. Interestingly enough, the pattern of GABAergic neurons is not only conserved between C. elegans and C. briggsy, but also C. elegans and a whole variety of much more distantly related nematodes. So it's a fairly conserved trait, the number and the relative positioning of these neurons. We can recapitulate the endogenous pattern of expression of UNC47 in C. elegans if we fuse 1 kb or so of DNA immediately upstream of the gene to the GFP. And this is what's shown here. So these are the 26 cells that are driven, or expression which is driven by the roughly 1 kb promoter of UNC47. Same story in C. briggsy. A 1 kb fragment upstream of C. briggsy UNC47 also animates a pattern that is these 26 cells. All right, so here we have a trait that can be meaningfully considered conserved because you have 2 species, conserved number of cells, conserved expression pattern, and seemingly conserved regulatory elements because the same bit of DNA upstream of UNC47 in elegans and UNC47 in briggsy animates the correct expression. So what's the deal? What are the mechanisms? I'm asking this as a molecular geneticist. What are the mechanisms that are responsible for this conservation? Well, the first place that all of us now go is genome browsers. So let's compare if, in fact, there's been conservation of UNC47 regulation between elegans and briggsy. We should be able to go to the genome. We should be able to interrogate the sequences that drive this expression and find similarities and differences. Very good. So what I'm showing you here is a visual representation of alignment of C. elegans DNA immediately upstream of UNC47 and that of C. briggsy, C. riemanni, C. brenneri. Now, some of you perhaps have used this program, but basically the idea is every time you see a set of pink peaks, that is where you have stretches of conservation. The scale is here 1.4 kb. ATG of UNC47 is right here. ATG is going this way. And the gene immediately upstream of ATG, called EPAC1, is going that way. Right? So you have a divergent pair of genes separated by roughly 1.5 kb of DNA. Now, what immediately strikes you is that the most proximal portion immediately upstream of the ATG is highly conserved. Okay? So you have something on the order of 250 nucleotides. And we've looked at it in a variety of different ways. One way in which you can look at it is ask what fraction of this sequence is covered in 10 nucleotides that are identical between C. elegans and C. briggsy, for example. And in this frame of 250 nucleotides, it's on the order of 30 to 40%, whereas in the remaining sequence over here, it's basically zilch. Okay? So whether or not there's something here that we can detect, we're going to address in a few moments, but at least what is clear is that the profiles of conservation here are vastly different. Very well. Remember, the patterns of expression are conserved, and so what we're thinking is perhaps conserved bits of promoters are sufficient to recapitulate conserved patterns of expression. We can test that experimentally by comparing the function of a proximal promoter, a short promoter, that just encompasses this conserved sequence up to this boundary, and the full-length promoter that extends all the way to the next gene upstream. Okay? So let's look at them and let's compare, let's test whether or not they're functionally equivalent. And so to the first glance and the first qualitative level of analysis, the answer is yes. Now I'm fudging here a little. In fact, I've cut and pasted the same picture here to confuse you a little bit. But to a naked eye, the pictures look pretty similar. But we started noticing, we started really looking at them a little bit more, that there are subtle differences. And so what we were unable to sort of look and judge what those differences were. Therefore, we decided to really quantify, put some meat on the bones and really do the numbers. Okay? So what we're going to do is we're going to measure fluorescence in individual cells in large populations of these animals, and we're trying to quantify similarities and differences. Okay? So there's a variety of ways in which you can represent these data. One way to represent these data is to do the following. Let's take measurements from 6 cells in the ventral cord. We have reasons to believe that they're regulated by the same genetic mechanisms, and derive an average amount of expression in an average representative cell. So we take 6 measurements and average them, and then we compare them to expression level in the cell called DVB, the postanal neuron. Okay? And so we can ask a question. For example, let's take a ratio of DVB expression to ventral cord expression. And what I'm showing you here are the actual data showing on this scale the ratio of DVB expression level to VNC, or ventral cord expression level, and individual dots are individual values. Now, full-length promoter, that 1.3 piece of DNA that extended all the way to the gene upstream, 2 things are notable. First, the mean is right around 1, okay? Meaning that on average, DVB expresses about as brightly as cells in the ventral cord. And second, the scatter is pretty tight, and it's definitely a normal distribution, meaning that there's some noise in the system, but there isn't a lot, and it's normally distributed. Very good. Now, let's look at the proximal promoter. The proximal promoter, again, is just the conserved portion of that promoter, the 250 nucleotides immediately upstream of the ATG. Now, remember, the patterns, qualitatively anyway, were similar. What you see is 2 things. First, there's a substantial shift, downshift, in the mean expression level of DVB relative to the ventral cord. Okay? So DVB is half as bright now as cells in the ventral cord. But second, it's very significant, variance is greatly increased, which is counterintuitive because anytime you decrease the mean, you are expecting to see a lower variance. That's not what we see. In fact, we see quite the opposite. Now, it's even worse than it looks, because just to show you this slide, I had to cut off a number of points that are located at 5, 10, and 15, literally off the charts. And, of course, these differences are highly significant. So what's going on? Well, before I tell you what's going on, there is another way in which we can quantify similarities and differences between these promoters. Right? I told you that the transgene and the gene itself are expressed in 19 cells in the ventral cord. And so what we can do, we can compute a very simple statistic. We can ask, in a population of animals, how many animals express in how many cells. All right? And those data are represented here in this distribution. Right? So you have a fraction of animals in the population that express in 19 cells, 18 cells, 17 cells, 16 cells, and so on. And there are 2 distributions. The black distribution represents those animals carrying the long promoter, and the white distribution, those animals carrying the short promoter. And the results are unequivocal. The short promoter is a lot less consistent. So more animals express in fewer cells. fewer animals carrying the short promoter expressed in all 19 or 18 cells. Very good. Okay, now this is a comparison of just two species, and perhaps there are some pitfalls to this comparison. Are we really capturing the picture? Is it really a consistent phenomenon that the short promoter is different from the long promoter? So what we did was to test two other relatives, C. Riemanni and C. brenneri, for essentially the same phenotype. We compared the function of the long promoter to the function of the short promoter. And results were exactly the same. Now, again, we're comparing either promoters derived from C. brenneri or from C. Riemanni, and we're looking at either a full-length promoter or a short promoter. In both cases, the long promoter is expressed consistently in many more animals in all cells or nearly all cells. So again, the long promoter is always better, regardless of whether it's from C. elegans, from C. briggsy, C. brenneri, or C. Riemanni. So that's a consistent phenotype. Now, as we were conducting these experiments, we were trying very hard to keep the environment as consistent as possible. All of these worms were raised in conditions that we tried to control the best we can, same temperature, same everything. Now, if there's so much variation, then perhaps we can introduce extra environmental variables and drive the differences between these strains further apart and really test the environmental sensitivity of these promoters. And so that's what we did. Now, what I'm showing you here, and you're going to see a few slides like this, so let me go ahead and explain the structure of one of these graphs. I'm plotting on the x-axis the log intensity of average amount of expression in D-type neurons in the ventral cord. And on the y-axis, again, on the log scale, amount of fluorescence in DVB. All right? And there are two types of circles here, light blues and red, and they represent two different populations. So we took worms of the same genotype carrying the same promoter and split them into two populations. Some were reared for generations at 15 degrees, which is the lowest at which these worms can be reasonably maintained, and others at 26, which is the highest temperature these worms would tolerate. So when you look at animals that carry the long C. elegans promoter, that is the conserved bit plus the distal domain, you see two things. First, the distribution is fairly tight, and the blue and the red substantially overlap. And what that means is basically all animals do basically the same thing with regard to their expression. And it doesn't really matter whether they're in a cold condition or the hot condition. Animals that carry just the proximal portion of the promoter, just a conserved portion of the promoter that gives you essentially the correct expression pattern, absolutely don't do that. These two distributions are very different. This distribution is a whole lot more variable, and particularly variable in the blue domain. That is, effectively, you can think of these worms as being cold sensitive. Under conditions of cold stress, variance of expression grows quite considerably. Same exact result is seen when we look at transgenes derived not from elegans, but in this case from briggsy. Again, we're comparing the long promoter, that is the conserved plus non-conserved, to just a bit that is conserved. This distribution is fairly tight. Blue and red, for the most part, overlap, whereas this goes away completely when you just look at the short promoter. Okay? And so it's not as if the short promoter is somehow broken, because at least in high temperatures it works reasonably well, but it's cold sensitive. And so we're starting to put together a picture where in the promoter of UNC47, we can define two distinct topological domains. One is highly conserved, the other is not at all. One drives either all or nearly all endogenous expression pattern. The distal portion on its own is not capable of sustaining a pattern. However, the proximal promoter, even though it drives the majority of expression pattern, is not in itself robust. The expression that it generates is not robust. It is sensitive to environmental conditions, whereas the full-length promoter, that is the addition of this distal bit to the proximal, endows this promoter with robustness. Now, I'm giving talks, right? So I have to find a visual metaphor to explain to you what I think we see. And of course, you do a Google search, you do image Google search for words robustness. And the first thing that came up was this. And the way I read this is ingredients for life, are ridiculously robust. Ha ha. Well, I couldn't stop there, and I found this cartoon. So this actually seems like a ha-ha cartoon because it's Dilbert, but first, I don't find Dilbert very funny, and second, I think there's a lot of real sense in this cartoon. Now, let me give you a background. They were tasked at the company that they work at to come up with a robust product. And what's the first thing to do when you're given a task that you know nothing about? You hold a meeting. So they're having an animated meeting, and they're discussing the meaning of the word robust. And this guy, Dilbert, I think, says, I think robust means it has a lot of features. And this guy, whoever he is exasperated and says, no, it means sturdy. So when I first introduced the term robust here, I meant it as sturdy. That is, you change the environment or between different animals, expression is basically the same. So that is robust to me. But I actually think Dilbert's onto something despite being a two-dimensional cartoon character. Having lots of characters really helps you to be sturdy. Now, that's a proposition that I will try to illustrate with a few experiments that I'm going to tell you about in a minute. All right, so let's try to get down to the meat of this business. I told you that this distal portion of the promoter had something to do with endowing the short portion of the promoter that in itself is not particularly robust with this magical robustness. How does it do it? Well, so we can examine it from several different perspectives. And for example, we can ask whether or not the distal portion of the promoter, the robustness endowing portion of the promoter, somehow talks in a sequence-specific way to the proximal portion. We can ask in that long 1 kb fragment of DNA, which doesn't seem to be conserved, is there anything? Are there hidden messages? Can we find hidden messages? Are there motifs that we don't know how to identify, but perhaps we can see them if we really look very hard? And is it just these sequences that confer robustness, or is it anything? All right, so let's look at the first question, right? Remember, when we took the briggsy promoter, the full-length briggsy promoter, it was, by our definition, fairly robust. A fairly tight cluster, blue and red, overlapping considerably. Now let's swap the distal portion of the briggsy promoter for the distal portion of the elegans promoter. Well, the two are basically the same. So remember, again, there is no sequence similarity to the level that we can detect easily between this sequence and that sequence. They're unrelated so far as we can tell. And yet they work quite fine. Let's examine the difficult issue of hidden messages, of little motifs that are dispersed. Well, so it's a very hard problem, it turns out, right? Because if you compare any two 1 kb sequences, you will always find some match, right? After all, it's only 4 nucleotides of DNA, and particularly if you have a biased composition, then having matches is really not surprising at all. And I was trying to be adamant about there being a really sharp boundary at 254. Well, there is a really sharp boundary at 254, and within the first 250 nucleotides, there's a great deal of conservation. There's a great deal less further beyond. But it's not to say that there's absolutely nothing. So there are a couple of motifs that were located just outside the 254, and those motifs could be found in elegans at briggsy and Romania. As you can see, they're fairly short, and they're roughly 70 to 100 nucleotides upstream. So now we're comparing a long promoter, that is the conserved portion, the 254 conserved portion plus that distal bit, to the conserved portion that also includes all of these and a little bit of surrounding DNA. And what you can see is that whereas this is clearly worse than that, that's the p-value that shows the difference, this is a hell of a lot better than it could have been, right? Just a short promoter alone is worse than this. So adding these sequences and some sequence around makes it better, but not all the way. Well, let's ask whether it's any piece of DNA. And so for that, I didn't want to do this experiment. In fact, I didn't do any of these experiments, but I'll tell you about that later. But I didn't want this experiment done. I didn't want to pay for this experiment, as it were. And I have a graduate student who said, I want to do this experiment. And I said, well, that's a stupid experiment. Don't do it. I'm the boss. I know what to do. And of course, she did it anyway. And it turns out that it paid in spades. And the reason it paid in spades is because she said, what is a piece of DNA that is most unlike in terms of its expression pattern compared to UNC47? Well, it turns out UNC15 is a pretty good one, because whereas UNC47 is expressed in 26 GABAergic neurons, UNC15 is expressed in somatic muscle. There's zero overlap between these two. These two genes are totally not related in any way whatsoever. And she just picked a 1 kb fragment of DNA someplace upstream of UNC15. Look at these two. That's pretty remarkable. So a piece of DNA from a totally different locus basically confers almost as good a robustness upon you as your own robustifying piece of DNA. So we've repeated this now several times with other pieces of DNA from other unrelated genes, and that's basically what we see. When we take a piece of DNA upstream of an active C. elegans gene, it robustifies. All right, now I have to tell you a little bit more now. This is where we're getting into nitty-gritty details of these experiments, quite how these experiments are done, all right? So we're using standard constructs everybody in the field uses to make transgenics. They contain GFP. And the difference between the full-length promoter construct and the short promoter construct is just the position of the vector. It's the same vector, right? So it either has this bit of DNA or it doesn't. Now, when we make these swap constructs where we exchange the distal bit for the proximal, exchange this distal bit of UNC47 for the distal bit of UNC15, we're just cleanly replacing this piece of DNA with that piece of DNA. All right? So there's got to be something in common between this piece of DNA and that piece of DNA that is not a feature of the vector. Okay? So what is it? Clearly it couldn't be sequence conservation, because I started out saying even between UNC47 distal pieces of DNA there was no sequence conservation that we can see. Well, clearly there's no sequence conservation between UNC15 and UNC47 distal pieces of DNA. What is there? Well, we have no idea, but the first thing that springs to mind when we think about it, and we're in the business of sequence analysis after all, is nucleotide composition. So let's look at general features of nucleotide composition. Single nucleotides are not particularly informative. Let's look at dinucleotides. And when we've done that, every single piece of DNA that was able to confer robustness on this proximal UNC47 promoter had a peculiar feature. That is, they were enriched for AA, double T nucleotides, dinucleotides, and substantially depleted for CCGG or GCCG. All right? I'm sure half of you in the audience, maybe 2/3, and maybe all of you are thinking, this smells like chromatin. So let's consider the chromatin organization of the UNC47 locus. All right, so this is the UNC47 locus. This is where the ATG is. This is the short promoter. That's the long promoter. That's the gene immediately upstream. That's 1.5 kb between the two. Now what you're seeing here is a nucleosome occupancy profile in which areas above the line and shown in yellow are enriched over genome-wide average, and areas in blue and below the line are depauperate for nucleosomes. And what I find absolutely striking is this nearly perfect coincidence between the boundary of high conservation and the onset of a dip in nucleosome coverage. Now, it turns out that genome-wide, in these blue areas of low nucleosome occupancy, 3 particular trinucleotides are preferentially found. And when we analyze our robustness rescuing sequences, they are enriched for these 3 trinucleotides and depauperate for nucleotides that are present in sequences that have higher nucleosome occupancy. All right. So right around the time when we were finishing all this, 2 papers came out in quick succession talking about very similar mechanisms by which gene expression can be made robust. And basically the idea is this. There are these things called shadow enhancers, where a gene is driven by a particular enhancer, but there's another sort of secondary auxiliary enhancer that can drive in the same pattern And if you're just looking for GFP expression or beta-gal or whatever reporter gene, the two are equivalent. But if you make mutations that abolish one enhancer or the other, and then stress the animals, expression and phenotype of the genes phenotypes caused by these genes are made less robust. Now, ours is a very different story. We don't have shadow enhancers because the distal piece of DNA in itself is unable to drive expression. And remember, we can replace UNC47's distal bit of DNA with that of UNC25 or UNC15. So we have, what it seems, a different mechanism by which gene expression can be made robust, and perhaps it is mediated by decreased nucleosome occupancy. But as an evolutionary biologist, what I'm particularly excited about is that what we've been able to do was to connect functional constraint, that is, whether or not a sequence, in this case, drives a pattern, or in this case, robustifies it through whatever mechanism, and its rate of evolution. Because one of the big questions in the field is trying to understand why is it that some portions of the genome evolve fast and others evolve slow. And the reason this may be evolving very fast is because, again, it is explained by the experiments I've been telling you about. UNC15 distal portion can rescue robustness quite sufficiently. So maybe the requirement on this piece of DNA isn't just to conserve some sort of primary sequence, but to basically maintain di and trinucleotide composition that is overall consistent with lower nucleosome occupancy. All right, so it does appear from this analysis that from the common ancestor of C. elegans and C. briggsy and these other nematodes that I've discussed, a promoter of UNC47 can be divided in these two domains. One is driving the pattern, and it's pretty well conserved. The other is not so well conserved, and it's responsible for robustness, and the function or the changes have primarily occurred over here. So you can ask a question. Have there been functional consequences to those changes that have clearly accumulated in the distal portion of the promoter? Because remember, after all, when you compare promoters of C. elegans and C. briggsy UNC47, you can absolutely align the proximal portions, but you absolutely can't align distal portions. So were there functional consequences to these mutations clearly having accumulated in the distal portion of the UNC47 promoter? And the answer is yes. And the reason we know it is because if we take the UNC47 promoter from briggsy and stick it into elegans, we first, of course, see the recapitulation of the endogenous gene expression, but we also see ectopic expression. We see that ectopic expression is turned on in several neurons, but I'm going to discuss just two here. They're called SDQR and SDQL. And this sort of picture really illustrates why C. elegans, for our purposes, is a wonderful model organism, because I can tell you what these cells are, and it can be very specific about it. Now, it so happens that we've tested whether or not UNC47 promoter from Riemanni or Brenneri also get to be expressed in these SDQR and SDQL, and the answer is basically not. It's not absolutely not, but it's very, 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 very low. Right? So essentially, it's a property of briggsy DNA when put into elegans to be expressed ectopically in these two neurons. All right? So yes, okay. So don't read all of this table. I promise to come back to it again. But what we've now done is a complete series of cis-trans transgenesis, right? So we've put the long promoter of C. briggsy into both elegans and briggsy, the short promoter from C. briggsy into both elegans and briggsy, the long promoter of elegans into both elegans and briggsy, and the short promoter, okay? And so by analyzing different rows and columns of this table, we can draw some conclusions about what's happening. All right, so lesson number one. Lesson number one is that long promoters are always more ectopically expressed than the short ones, because these numbers are the fractions of individuals in populations that either ectopically express in R, SDQR, or SDQL. All right? And so in every comparison shown here with a red arrow, that's where we have substantial numbers of ectopically expressing animals. Every time you compare, a long promoter is always more ectopically expressed than the short. Okay. Now, remember we did those funny experiments, or rather, the student whom I told not to do these experiments did these funny experiments with distal DNA from UNC15 and UNC25. Well, it turns out that those promoters, those chimeric promoters that were made robust by the addition of this DNA, are expressing basically as well as the very worst overexpressors. So that's lesson number one. Long is more ectopically expressed than short. Okay? Well, that's an easy problem, right? So we have a piece of DNA, we have ectopic expression. We know that it's something that's resident, whatever that is resident somewhere in the distal portion of the promoter. So let's just find what it is. And so we've done so. So this is an abbreviated promoter deletion series where we took steps in deleting, progressively deleting distal portions of the promoter until we lost all the ectopic expression. And so what I'm doing here, I'm summarizing, and again, these numbers refer to fractions of individuals that ectopically express either in SDQR or in SDQL. And what you can see is that you can lob off a very substantial portion of this promoter and basically not lose any of the ectopic expression. But then a fairly small cut makes you lose a little, then another cut makes you lose a little more, and then tiny little cuts like this cut from 266 to 254 lose you some more. But even the shortest, highly conserved portion of the promoter still has a substantial amount of ectopic expression. So the lessons from this are that there are multiple sites, and the individual contribution of these multiple sites is actually fairly subtle. Because after all, you're just dropping 10-20% of ectopic expression in one of the two ectopically expressing cells. I mean, this is as mild a phenotype as you're ever going to come across. All right, so what we're thinking about is a model where this congregation of sites, these sites are somehow talking to the basal promoter and together collectively affect ectopic expression. Lesson number two that you can glean from this table is that C. briggsy DNA is always more ectopically expressed than C. elegans DNA. Okay? And it's not only seen in C. elegans, which you could argue is, well, that's a trans-species comparison, but it's also in its own self. C. briggsy DNA is more ectopically expressed. Consider, for example, long briggsy to long elegans, or long briggsy to long elegans, or short briggsy to short elegans, short briggsy to short elegans. So briggsy DNA has this magical property that regardless of whether it's in elegans or in briggsy, it is more ectopically expressed. Conversely, it is the C. elegans trans that is promoting more ectopic expression than C. briggsy trans. And again, it's these comparisons that allow us to conclude that. All right, that's another relatively straightforward molecular biology problem. If it's C. elegans trans, we should be able to map what that C. elegans trans is. And so we took a candidate gene approach and asked, what are the transcription factors that are expressed in these two cells, SDQR and SDQL? Because after all, by definition, it has to be a transcription factor expressed in those cells in order for ectopic expression to be regulated by it. One of the genes that floated to the very top of the list is this transcription factor called AHR1. And AHR1 has a distinction of not only being expressed in these two cells and a few others, but also in two cells in the head in which its expression pattern overlaps with the endogenous pattern of UNC47, suggesting perhaps that UNC47, at least in a portion of its expression pattern, may be a legitimate target of AHR1. Okay? Now, the question is, what happens to the ectopic expression in the background of AHR1 mutation? Now, I wouldn't be telling you if the result was anything but this. In the background of AHR1 mutation, you completely lose ectopic expression. So these are the animals carrying the briggsy promoter. Otherwise would have been expressed in all or nearly all animals. Ectopic expression is completely gone. Now, the cells are there. So the cell bodies are there. It's not that the cells aren't there. It's that the ectopic expression is no longer present. Very good. The proximal portion of the promoter of UNC47 contains what seems like a very good consensus site for AHR1 binding. Let's abrogate it and see what happens to expression. So we've done so. In the promoter where AHR1 site is intact, all or nearly all individuals expressing at least one and oftentimes in both SDQR and SDQL. When you site mutagenize the site expression, ectopic expression goes away completely. So not only AHR1 does this dirty business, but it does this dirty business via this conserved site. And so the model has now morphed into an interaction of a number of motifs in the distal portion of the promoter, somehow talking to this conserved AHR1 site, altogether resulting in the ectopic expression that we observe. All right. So there have been functional differences. There have been functional differences that have accumulated between C. elegans and C. briggsy. And yet, endogenous pattern of expression of UNC47 are the same between elegans and briggsy. So what this strongly suggests is some sort of a compensatory evolution. That is, changes have accumulated in both lineages, but in both lineages they have been subtly adjusted to result in the overall conserved pattern of expression, or a pattern of expression that to us appears to be conserved. Now, final time I will invoke this table. Again, remember, we're moving either long or short briggsy DNA into either elegans or briggsy, and then elegans DNA also into elegans and briggsy. And to summarize crudely the results that we see is that briggsy DNA when in elegans is highly overexpressed. Briggsy DNA when in briggsy or elegans when in elegans is somewhat overexpressed. But elegans DNA in briggsy is not. And so this is not just some dysgenesis. You move the DNA from one species into another, and all of a sudden it doesn't know what to do. This is a telltale sign of opposing forces of compensatory evolution meeting someplace in the middle. And that is why you see high here and low here. So in genetic terms, this is very much like transgressive segregation, where you have offspring be more divergent in phenotypes than the parental types. And so the model that we have at the moment is that, remember I was telling you about briggsy having strong cis, that is, cis DNA from briggsy being more ectopically expressed than cis DNA of elegans. That strong briggsy cis is compensated by weak trans, or vice versa. Weak, in converse, weak C. elegans cis is compensated by strong trans, overall resulting in a moderate level of expression. But when you move a strong cis in the environment of a strong trans, which is the case when you move C. briggsy DNA into C. elegans, you massively overexpress. And the converse is true in the reciprocal transgenic experiment, where you combine the weak cis with weak trans, where you see hardly any overexpression. So what could be the cause of this weak trans or strong trans? And it appears that it's the level of AHR1 expression. AHR1 is just lowly expressed in briggsy. The overall level of AHR1 expression seems to be lower. And so the funny bit is that even though what we seem to have here is coevolution between cis and trans, compensating a weak cis with a strong trans, in reality, the trans change is actually cis itself, because it's the overall level of expression that seems to be different. And so what I'm going to propose coming out of this is this notion that perhaps we're not looking at conserved traits when we're thinking conserved traits. Conserved would imply that the trait hasn't really changed. What hasn't changed is the appearance of the trait to us. But the molecular mechanisms that determine when, where, and how these genes are expressed have clearly changed. We've been able to map these things down to actions of individual cis-regulatory elements and transcription factors, and yet the overall pattern of gene expression has remained static. And so perhaps instead of thinking of these gene expression patterns as being conserved, we ought to be thinking about them as canalized or buffered to changes. They are in constant flux. They're in constant evolutionary, co-evolutionary turnover that at the end result in indistinguishable to our eye patterns of gene expression, but that belies a great difference in the weight grade, a little bit of difference in the way how these genes are regulated. So there has been functional change. It is documentable. We can map it very specifically, and yet the overall pattern stays the same. I have a little bit of time left, right? Right. Okay, so let's think about this. So I spoke for, 45 minutes about basically one gene. Are rules the same or different for different kinds of genes? And that is the question that the lab is currently very much engaged in. Okay, so I don't know, 7 years ago or so, when I was still in Boston, Gary and I published a paper in which I did what now in retrospect seems like a stupid experiment. And that was go to literature, identify fly genes whose cis-regulatory elements have been well characterized functionally. That is, you know that this piece of DNA, when in the fly, drives expression of a particular reporter gene in those cells, okay? And I fused a bunch of those to GFP, stuck them in the worms, and I was expecting a miracle, and a miracle didn't happen, and so these were not expressed. But they were not expressed in one of two funny ways. They were either not expressed, and we can make transgenics, so it wasn't that, or they were totally expressed in wrong patterns. Neuronal genes would be expressed in the muscle genes in neurons, and so it was a total mishmash, clearly not betraying conservation of underlying cis-regulatory circuitry. All right, so the conclusion from that was that cis elements from flies cannot be understood by the transregulatory machinery of worms. Well, that's true for this particular set of genes. And there was a bias as to how these genes were picked. I was picking sexy genes, and sexy genes are those that are regulators of development expressed in very specific places and times. And so perhaps it was that unconscious bias of which genes to look at that determined the outcome of this experiment. And so there are two classes of genes that we have completely omitted from that survey. Ones were boring housekeeping genes ubiquitously expressed, your metabolic enzymes and ribosomal proteins, okay? Maybe their promoters are different. Maybe their promoters evolve under different regimes. Another group of genes were inducible genes, and it is those two that I will now speak about. So here's an example, and if you're not seeing anything major, it's good because you're not supposed to. So what we've done was to, again, go in the literature, look up papers that said this piece of DNA upstream of a highly conserved ubiquitous gene in the fly drives its expression pattern, blah, blah, blah. We fused it to GFP, stuck it in the worm, and what we see is nothing. So we've intentionally overexposed these to a great extent. We see fluorescence in the gut in all of them, and we see no specific pattern of expression. Clearly not ubiquitous pattern of expression. So promoters of ubiquitously expressed genes, at least these four that we've tried, don't seem to be functionally any more conserved than promoters of tissue-specific genes that I spoke about just a moment ago. Not so for heat shock genes, okay? So here's the experiment. Again, went in the literature, took promoters of heat shock genes, fused them to reporter genes, and then induced heat shock, and then monitored by QRT-PCR levels of induction, okay? Black is, okay, that's the promoter of HSP26 from Drosophila. That's the promoter of, or these results, this panel reflects melanogaster HSP70. Black is uninduced, that is flat at 1, that is no change over level before the onset of heat shock. Red is heat shock. This Drosophila promoter, when in elegans, is induced to roughly 4-fold, and that is ballpark comparable to the level of induction that you observe in the endogenous situation, that is in the fly. HSP70 shoots up to roughly 100-fold within, less than 2 hours, and that again is ballpark comparable to what we see in Drosophila. And so we've tried 2 more promoters, one from a fly and one from human, of other heat shock genes, and we see the same thing. So what we see is that contrary to what we saw with tissue-specific transcription, tissue-specific genes and ubiquitously expressed genes, promoters of heat shock genes from flies seem to be, at least to some extent, interpretable in the worm. Now, the levels are off, the patterns are funny, but the ability to be induced seems to be understood, or the need to induce such a promoter seems to be understood by C. elegans. Why is that? Well, I can't really offer you an answer. I don't know if there is an answer. But I can offer you three ideas that are certainly not mutually exclusive, but perhaps together they start painting a picture of how cis-regulatory elements change over time. Well, one could be that really this dichotomy between inducible genes like heat shock promoters and those others is an illusion. Because what we're asking are different questions. When I'm asking for a promoter of a tissue-specific gene to be conserved, what I'm really asking is for it to be turned on in the right cells at the right time. What I'm asking of a heat shock promoter is to be just responsive to heat shock. I'm not asking for pattern. I'm asking for ability to be induced. And that may be fundamentally different. There's also some notion that HSF, that is the transcription factor that binds to these promoters and activates expression, may be unusually conserved. Now, that may be so, may not be so. It's very difficult to even know how to experimentally address this. There are other highly functionally conserved transcription factors. But perhaps this plays a role as well. What I think is, and again, I want to stress that these are not necessarily mutually exclusive. What I really want to stress is a possibility that maybe this reflects a difference in wiring logic of gene control between heat shock genes and tissue-specific genes. Whereas for a tissue-specific gene to be expressed in a complex pattern of cells and times, what you need is integration of a large number of signals coming from upstream. This has to feed in, that has to feed in, 100 different other things have to happen just right. Not so, or not quite so, for heat shock promoters. Heat shock promoters are heat shock promoters because they have HSE sequences that are recognition motifs for HSF. And if HSF is functionally conserved, which it does seem to be, it would seem logical that it would bind to the same sequences. And if that's the case, you already have your HSCs in your promoter. HSF's around. Upon induction of heat stress, you bind, you activate expression. So perhaps this really does account for this divide between tissue-specific and inducible genes. So let me round out by highlighting some of the things that I told you about, and I will leave time for some questions, and if there's more time and interest, I'll tell you what we're doing at the moment. So what I told you about is a story of fairly detailed dissection of promoter of gene UNC47 in which there's a highly conserved and a highly divergent portion. And highly conserved is in itself sufficient to give you basically the right pattern, but perhaps not quite. But certainly it doesn't make its expression more robust. And what these different constraints, that is, this bit makes it be robust, this bit drives the pattern, that perhaps explains the difference in rates of evolution that we observe. We've analyzed the functional divergence of this regulatory system, that is, UNC47 and transcription factors that bind it between C. elegans and C. briggsy. And what we found is that divergence is due to a large number of individual sites that have functionally changed. And we've identified specific trans regulators that have concomitantly changed. And perhaps the funniest thing about those trans regulators that have functionally changed is that the trans change really isn't trans change, but a cis change. I further suggested a hypothesis that perhaps a lot of the traits that appear to us to be conserved because worms look the same or flies look the same or what have you, really is an illusion of divergence that's been compensated. Rapid rounds of change and calm on the waters. Finally, I told you a story about how heat shock promoters do seem to be a little different than everybody else in that their function, their ability to be induced by heat shock does seem to be more highly conserved or retained for longer periods of time than the ability of other transcription factors, other cis-regulatory elements to be functionally conserved over that same distance. As I mentioned, none of this work was really done by me. It was started in the lab by a postdoc, Antoine Barrier, and was then very ably helped by a very talented graduate student, Casey Gordon. She's the one doing those experiments that I explicitly forbade. Some of the work that I told you about is that of Hands of Paul Wang, Zheng her, Aaron Epperson, and others. This is who pays our bills. If you want more, there's more, and I'm happy to take your questions. Yes?
Since you're starting to look very carefully at these promoters, are you seeing or suspecting any effects due to the fact that, at least in C. elegans, if you do it the traditional way, you're getting long tandem arrays of these genes not in anything at all?
There's $5 in this question for you. This is a very important concern that we prefer not to take outside the worm meeting, but since I was asked, I'm going to say. Right, so traditionally, transgenesis in the worm is done in the following way. You take a piece of DNA, it's a vector. Sometimes people do PCR fusion, and you just massively inject. And what forms is episomes. Very rarely do you have spontaneous integration. Sometimes people integrate. But at any rate, when you do it this conventional old school way, you have a tandem of 5, 10, 100, several hundred concatenarized GFP promoter fusions, right? So we're very much concerned about this. And so we went an extra mile and decided not only are we going to make it single copy or low copy, we're going to make it site integrated. Okay? So what we did was site specifically recombined into the same docking site in the genome of C. into a known same docking site in the genome of C. elegans, either the long promoter plus GFP or the short promoter plus GFP. And what I'm showing you here is a comparison of the long promoter in dark blue and the short promoter in light blue. And remember, we're counting the number of cells that express that construct. Light blue is significantly different than dark blue. Now, this is something that I should have known ahead of time. This is something that is taught in every science class around the world. Sometimes an experimental tool that is more precise is not necessarily better. This is actually, for our purposes, a worse experimental tool. And the reason it's a worse experimental tool is because the level of expression is very, very low. This is a single copy integrant, and so we cannot measure these very well. There's a certain amount of imprecision in when we take these fluorescence measurements, and that we cannot do anything about. This level being so low, imagine there's a mean difference of this versus this. And if you experimental variances like that, the two distributions are going to overlap. So they kind of do overlap here. We see a trend, and if you plot the dots and you build a regression, it's in the right direction. But the amount of experimental variance that is just inherent to these experiments cannot be reduced. We cannot reduce it any further. Maybe it can be. But this is the case where we actually had to go with a worse, or less precise experimental tool because it allowed us to do so. But the concordance in what we can measure using this and non-integrated strains assures us that we're onto something real. Yes?
So I'm wondering, one of the elements that they have in for software are boundary elements.
Yeah.
So do these things define boundary elements?
It's yeah, the question was, if I understand it correctly, is some functionality of this robustness promoting sequence a boundary element? I cannot answer this. We're definitely looking at something related, whether or not you see more of such sequences between genes that are divergently transcribed as opposed to convergently transcribed, because that might be one of the predictions. You need to, if you have two promoters, one facing this way and expressed in the gut and not expressed in neurons or whatever, you may want to separate them. We don't know. We don't really know boundary elements very well. There is a set of data now of the transcription, a factor that binds to these elements that is now escaping me, the four-letter word, as it were. CTCF, thank you very much. CTCF. So there's now a profile of binding of CTCF throughout the genome, and we're trying to look at it more carefully. But that's a great idea. I mean, something, some magic. I mean, I'm sure it's chromatin to some extent, but I don't quite know all about it. There are others? Yes, please.
So a couple of things. First of all,
Yeah.
Elements. Do you understand what those elements do?
No, no, a little bit.
And, is it, and so this sort of wild west of the promoter upstream, where it doesn't matter what's going on as long as there's no nucleus.
Okay, oh, I see what
The
you're.
evolutionary pressure is very different there than they are in these more conserved
Yeah.
places, because it seems to me that the cis and transacting battleground is still going to be taking place with conserved elements.
Probably. So here's what I cannot answer this, right? So very few promoters have been dissected to the level of detail that would be required to really answer your question. Here's what I can say. We did, for example, look whether or not anything that's 5 or 6 nucleotides or longer is found in both the in the proximal and in the distal. And the answer is nothing. Sorry, nothing remarkable. So it doesn't seem like the distal is just a redundant subset of what you already have conserved in the cis, okay? I'm sorry, not cis. In the distal, right, so the distal non-conserved isn't just harboring a whole lot of sites that are otherwise also found in the proximal. So that doesn't seem to be the case.
Okay, and the other question is the difference between usable promoters and housekeeping genes.
Yeah.
To me, it seems like, yes, there's a simplicity in what the e-shot but it's even more simple to imagine the selective pressure to keep a gene on in housekeeping genes.
Again, as I'm a huge aficionado of baseball, I call them like I see them. I don't explain them. I think you're probably right. Like, my intuition would be exactly like yours. If I had to guess, the promoters that control, it's actually not a guess, it's an informed guess. Promoters of ubiquitously expressed genes are a lot more complex than we want to say. So it's not like you're on all the time. It actually turns out that it's also partly piecemeal. That is, expression in these cells are controlled by these 2 sites, and expression in those cells by this, but there's a global control, and it's all very convoluted. And so, I think it's probably not, I wouldn't go as far as to say that ubiquitous promoters are simple in their organizational logic. In fact, I would suspect that they're not very much different than tissue-specific ones. It's just they are many tissue-specific ones on top of one another.
So in case of the HSP70 promoter,
Yeah.
HSP70 is expressed at some level.
Low, very, yeah.
The cells are not shown. Is
Right.
that ubiquitous expression?
Rick, is that ubiquitous expression? It is. Really low read-through, right?
Yeah, remember, this is 1 of 12 genes.
Right.
But I'm saying that is a type of ubiquitous expression.
But that we may not be seeing, right? So what we see is a massive on-ramp, right? Because we don't see any expression or not much expression of this promoter unless we're inducing, right?
I just had a question of clarification. So the AHR1 driven ectopic expression.
Yes.
It's a conserved binding site.
Exactly.
And is the AHR1 also involved in the expression in normal expression?
Ah, very good. Yeah. So I tried to skirt by this issue because it gets a little hairy, but we actually know the answer.
Why would the site be conserved?
Yeah. Well, so that I cannot answer. That we don't know. We have some ideas, but it's... right, so what Eric is asking is this. I mentioned that HR1 floated to the top of the list for 2 reasons. A, it was expressed in, well, it was a transcription factor, but A, it was expressed in these 2 cells, right, SDQR and SDQL, but also in these 2 cells in the head. And what Eric's asking, what is the endogenous function of the HR1 with respect to regulating UNC47 in those 2 cells, if indeed there is an endogenous function of this gene regulating UNC47? So it's not clear, right? The reason it's not clear is because in the mutant HR1, those cells don't die and they don't go away, they change fate. Okay, so these 4 cells are in a ring, and it's a pair, dorsal-ventral pair, and the left-right pair. And what happens is that you misspecify one as another. And you can only see it by projections. They're physically in the right places because they migrate to the right places. But they change their identity, and you can see that they change their identity by sending projections. So identity doesn't go away, and therefore ONC47 expression doesn't go away because the dorsal ventral pair still expresses ONC47, but via a different mechanism. So there's essentially two layers of complex control. Even if you take away HR1 in those cells, ONC47 is still expressed. But the reason it's expressed is not the left-right reason anymore, but the dorsal ventral reason now.
You also made a construct where you made a mutation.
Does it go away? More people. Okay, all right, fine, I'll tell you. So this is where it gets really complicated. In the context of the long promoter, if you mutagenize HR1, expression in the endogenous portion barely goes away. There are just two cells that we know to have looked at. Expression goes down some, from 90% to 75%, all right, if you believe that. But, right, so this promoter, I'm sorry, not this promoter is expressed in 90, and this goes to 75. But if you truncate it at just the conserved portion, gone. So what that suggests, of course, is that there are some complex redundancies that we're uncovering in a doubly sensitized background. First, you chop this bit off, and second, you've introduced a mutation. So it's a synthetic phenotype that you only see when you have two mutations, a distal deletion and a proximal point mutation. Right? So that's the best I can tell you, and the best way to summarize, really complicated, right?
Right, or, even if instead of having it in 99.99% of the time,
It's just 75, right? It's a phenotype. Exactly. That's exactly the argument, right?
We have one last, very short question. Otherwise, we can see the rest of your 95 slides.
It's only 78. Vina.
I was wondering if you know anything about the endogenous ONC47 itself.
Yeah.
Is it robustly expressed if you're including conditions?
Yeah, no, so this is a terrific question, and it's, as you know, extremely difficult to test. So, yes, I cannot answer this. We're sort of trying to look at it, but it's very, very hard because the differences are very subtle. And the only reason we're seeing is because we're massively amplifying the signal with a reporter gene, right? It's reproducible, but it's very subtle. So, you're looking at small variances perhaps on means that are very similar. They're non-overlapping distributions, but they're close.
But are the other diverging pathway genes also showing similar kinds of variants?
So we haven't looked at this level of detail. These are very, very laborious experiments, right? Because you have to stress the worms, and you have to do it in multiple strains because these are different arrays, and then you have to normalize this to that, and you have to take images of individual worms and individual cells, blah, 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 blah. It takes weeks to get one time series, basically. So we haven't looked. I suspect that all genes are somewhat robust, or many genes are somewhat robust. But that's about the extent that I can answer. So I don't really know.
With that, thank you very much, Ilya.